So I'm seeing the participants' numbers climbing, and we'll wait until it stops moving before I start your introduction.
Sounds good.
This is a really great turnout. I'm going to start talking now because we're a little past the hour. I'd like to welcome everyone to CCHA's Conservator's Corner. This is an irregular series that we offer, which gives us a chance to dig a little bit deeper into conservation subjects and the underlying theory as well as the practice. So it's really fun. We do not have it on a regular schedule, but if there's any topics anyone attending is interested in, we'd be happy to hear about that. And I'll mention right now as well that there will be a questionnaire that pops up at the very end. It's a little survey about what sort of programs you like and how we can offer better services to everyone in our audience. During the program, I'll ask you to put any questions into the chat, and then at the end, I'll be presenting them to Fleur, and I may organize them a little bit, but I hope to get to everyone and everyone's questions. We will be recording this webinar, and so we can send the video out to everyone if there's anything you need to look back to. And Fleur has also put together a written handout that will have references for further reading as well. I'd like to introduce our special guest today, Fleur van der Wood, who is currently the preventive conservator at the University of Arizona Special Collections. She has really initiated their program here that was before some very disparate actions. So what Fleur's doing is taking a global look at providing a really continuous care of the collections on all levels. So she's working through many different areas. This involves all types of training, planning, and I think most of all, communication. So her favorite part of the job is getting colleagues excited about finding bugs, speaking the truth about pressure sensitive tape, and getting to enjoy the beauty of a good book binding. Fleur studied at the University of Amsterdam Conservation Program, which I personally know is top-notch because one of her classmates was one of our paper conservation fellows. So I've seen how thoughtful and well-prepared the graduates of her program are. And because of our connection through Juliet, CCHA was fortunate enough to have Fleur come in person in 2018 to give us a presentation of her professional doctorate research project on dry cleaning and mold. It was incredibly informative, and the pictures you're going to see today are just so beautiful as well. I love looking at these, and I know you're going to find this talk both useful and enjoyable. So I will hand you over to Fleur now.
Thank you, Heather, for your kind introduction. I have really fond memories of visiting CCHA in 2018. And I want to thank CCHA for inviting me to speak today. And yeah, I'm really excited to share this presentation with a larger audience. So yeah, my name is Fleur van der Woude. I'm preventive conservator here at the Special Collections at University of Arizona in a new position that was created last year. We're based out of Tucson, Arizona, which is on the territories of indigenous people. Today, Arizona is home to 22 federally recognized tribes, with Tucson being home to the Tohono and Yaqui. And I want to acknowledge the deep injustices Native people have endured over centuries and are still living with. This place is their home, and I'm very much aware of that. So during my professional doctorate, I conducted a research project on mold growth on paper in collaboration with the Westerdijk Fungal Biodiversity Institute at the Royal Academy for Sciences, the Dutch governmental organization Cultural Heritage Agency, the Dutch National Archives, and the North Holland Archives. This was a 3-month research project focusing on fungal growth patterns on paper substrates and implications for dry cleaning effectiveness. And I'm really happy to share some of our results today and hopefully shed a bit more light on what fungal structures look like on paper and what it is we actually do when we dry clean fungal infested materials. I hope it inspires others to dive deeper into this topic. We need a lot more research on this topic. And I also hope to stimulate students to seek out multidisciplinary topics for their research and thesis projects. I found it a very enriching experience to venture into a different field, learn how to carry out research in a different way than I was used to, and build connections between the Dutch conservation field and indoor mycology fields. I assume everyone here has basic or practical knowledge of what fungi are and how they grow, but I'll go through a short refresher on the basics to make sure we're all on the same page. The genetics of fungi are closer to those of animals than those of plants, but they form their own kingdom in a classification of species. There's a great variety within the fungal kingdom. There are both microscopic and macroscopic life forms, and they vary greatly in structure, shape, colors, reproduction, and biology. Microscopic fungi can be divided in 2 main classes, yeasts and filamentous fungi. My research focused on filamentous fungi, which means they grow hyphae, but yeasts are prevalent in indoor environments and cultural heritage collections as well. So molds are virtually everywhere, as we know, and can colonize virtually any materials. If we're talking about indoor molds, and we are speaking of a large corpus of different species. There's unfortunately no typical profile for indoor fungi or for fungi found on cultural heritage collections. But broadly speaking, we know that the variation in fungal ecology between buildings is large. Fluctuation follows the local outdoor air and features of buildings and their occupants. I will not talk about specific growing conditions because I fear we'll run out of time, but it is mainly important to know that fungi can remain dormant and grow under a wide range of conditions, sometimes seemingly adverse ones, and they are capable of creating microclimates to a certain degree, for example, by adjusting the pH and water activity in their direct vicinity. They're a very successful kingdom of species. Mold growth typically starts with an activated spore, but interesting to know, mold species can also grow from other fungal structures, such as fragmented hyphae. The spore or fragment gets entrapped on a textured substrate and uses its ECM, extracellular matrix, to stick to it. If favorable conditions occur, the spore starts forming a germ tube, turning into a hyphae. More hyphae will start to grow from their tip outwards. These hyphae form the mycelium, on which more complex fungal structures grow, such as the fruiting bodies that produce new spores, and rhizoids, which are thicker rooting hyphae that grow into the substrate. Hyphae and their ECM are also used to excrete enzymes, such as a cellulase and cellulolytic species, and to transport water and oxygen from the substrate. The direction of growth of germ tubes and hyphae is essential for survival of the mold and is directed by the properties of the substrate through various mechanisms. And the photos that we're looking at right now, we'll see more in depth once we talk about the research. These are all photos that we made during our project. Another topic I want to mention before we move on is the distinction between active, inactive, and dead mold. Active mold is any mold that is actively growing, forming new structures, and metabolizing. Inactive mold is any mold that is currently not growing, but could start growing again in the future if the right conditions arise. Molds can be inactive or dormant for years or even decades, and under seemingly adverse conditions. Dead molds are any mold structures that are not alive anymore, for example, after treatment with gamma radiation, and will not be viable in the future. Whether alive, inactive, or dead, all mold structures pose risks for collection materials and for our health. And of course, if you come across active mold, fluffy, smelly, wet materials, you're dealing with an acute problem in your collection. So what does mold growth on paper look like? Paper is a heterogeneous material that provides mold with a plethora of nutrients, like for example, plant-based fibers, gelatin sizing, colorants, and even mineral fillers and potentially metal particles can be used. The surface properties of paper, such as its roughness, and the bulk properties, such as its porosity, are determined by the materials and the methods used in its production, such as sheet formation and finishing. Water is always present in pores between the paper fibers as well as inside the fibers, and molds seem to be able to use different forms of water present in paper. Mold spores can get into paper during its production and during use, storage, and treatment of the finished paper product. Hyphae can grow on the paper surface, but can also spread through the bulk of the paper, and hyphae of cellulolytic fungi can also grow into paper fibers. Methods of attachment of the mycelium to paper depends on multiple factors, including the species, the growth phase at a given moment, and the composition and condition of the paper. Mold causes changes in the substrate too, through the growth process and through metabolic activity. During their growth, molds excrete a variety of materials, including substances to improve adherence to their substrate, to adjust the relative humidity in their vicinity, and secondary metabolites, including many different pigments. Which substances are excreted depends on the species, the substrate, and conditions during fungal growth. And so there can be a great variety even within one fungal species. And here we are looking at some photos of that we made with a scanning electron microscope during our research project, and we'll look at these later in the presentation as well. But here on the right, you see one example of a fungus that seems to have grown into paper fibers and deposited spores in the fibers. So I'm sure everyone here is familiar with IPM best practices and the general steps in dealing with mold-infested paper-based cultural heritage materials. So we will not discuss these steps today, but I do want to emphasize that we always want to dry clean at some point in our treatment. Whether that is our only treatment step, whether it's done before working with ethanol or after gamma irradiation. It remains an important step to mitigate future risk of growth, further degradation of infested material, and to diminish health risks. As Florian stated in her book, Fungal Facts, this is the most critical stage in treating mold-infested objects. Conservators use various methods to mechanically remove mold structures. Various authors, like Michelson et al., state that a mycelium can be removed relatively easily from paper. Hanna Chepanowska, who obtained her doctorate in Lyon with research on the interaction between mold and paper substrates, expects that only superficial growth can be removed mechanically. Sequeira et al. Conducted an international questionnaire survey among paper and book conservators to inquire about their methods for dealing with molds, and found that many of us don't just clean the affected area, but the entire page when we come across mold growth. They commented that we can expect to be spreading the structures when we do so. So to our knowledge, neither of those statements had been tested in experiments before. In our research project, we wanted to extend the existing knowledge on the distribution and morphology of mold structures on paper and test the effectiveness of 3 widely used dry cleaning methods. And we looked at brushes, smoke sponges, and microfiber cloth, similar to the dust bunny microfiber cloth. We studied existing mold growth in books that were kept in quarantine spaces in various Dutch collections to describe growth patterns of these fungi. And we also inoculated samples let me see, going too fast. These are photos of materials from Dutch collections that we used. And here on the right, you see some of the samples in our variable pressure scanning electron microscope. And we also inoculated samples of 18th century laid paper with a gelatin sizing with 2 mold species from the Westerdijk fungal biodiversity collections. And we used Chaetomium globosum and Penicillium chrysogenum. Both strains we used have been collected in archival settings in the past. These samples were used to study growth patterns, but also for our dry cleaning tests. And that video was one of the techs at the Westerdijk Institute inoculating some of our first samples. We ended up using a different inoculation method. This gives you a little bit of an idea of what that looked like. And then both the existing and new mold growth were studied using microscopy and variable pressure scanning electron microscopy, which I will call SEM from here on out. So I'll first talk about the morphology of fungal structures that we observed in samples from Dutch collections of what we call existing mold growth. So all 7 samples that we studied showed hyphae and spores with a variety of ornamentation visible with SEM. Even within the same sample, there was a large variety visible. Longer hyphae were often fragmented. Hyphae that grew on the paper surface showed a diffused growth pattern attaching to paper fiber surfaces here and there. In 3 samples, we observed hyphae disappearing into the paper bulk, and in one of our samples, we could see hyphae deeper in the paper structure. We also noticed hyphae present higher above the paper surface. The spores were spread on top of the paper surface and fiber surfaces, but spores were also found deeper in the paper surface, nestled cozily in between fibers, and in 2 samples, we found spores in openings in paper fibers, as I showed earlier. We did not only sample paper that was obviously infested, we also sampled from parts of the pages that looked clean or non-infested with the naked eye. In all cases, these samples showed mycelium and spore distribution when studied with SEM. So here is an SEM photo, and you can see the magnification in the bottom for all these SEM photos. So this is times 350. And you can see different types of hyphae growing. You can see, or that have grown in the past. On the top left, you see beautifully ornamented hyphae that I actually used for my Christmas card in 2018. And you can see in between the paper fibers, somewhat bigger hyphae. And then, for example, with arrow number 2, you see very thin hyphae that are growing in between those. And then here is another example of somewhat thicker hyphae on the left, which some of them are ornamented. And these are probably perithecial hyphae, so that grow upwards from a rooting body. And on the right, you see a bundle of hyphae that are actually following a paper fiber surface, which is called thigmotropism. And we actually did not observe that in many samples, but this was one good example that we found. And you see, of course, a lot of small spores distributed along the hyphae and deeper in the structure. And here's an example of a fungus that seemed to have attacked the paper fibers a bit more. There was a lot of brittleness visible and hyphae and spores that seem to be deeper in the paper fibers. And arrow 1 is showing a bundle of spores. And this is probably a penicillium or a similar type of fungus that grows these like long strands of spores and a lot of spores on the hyphae, and we'll look at a similar type of growth later. And then under arrow 2, you can see a whole bundle of strands of spores. And here we see some spores deeper in the paper structure. And this was actually an area that with the naked eye looked clean and not infested. And here are some black spots that we're all familiar with formerly infested paper. And we think that we're looking at the kind of like base of former fruiting bodies, and we're looking at broken up rhizoidal hyphae that are growing into the paper. So this is one of the fungi that we inoculated our paper with, Chaetomium globosum. And I first want to show you kind of what these species look like and how they release their spores. These results really help me understand a little bit better what we're trying to remove when we're working with mold-infested paper. Chaetomium globosum is a cellulolytic mold. It's the most detected chaetomium in indoor environments. And it grows a round bottle-shaped fruiting body with a pore on top where its spores are released. This is a photo I made after sampling from one of our paper samples using sellotape and a blue stain. And you can see the bottom of the spore or of the fruiting bodies in the bottom of the photo with some of the rhizoidal hyphae. And then you see the perithecial hyphae that grow upwards, and you see some of the spores that are being released. And it was actually releasing some spores while I was making, or this is a different fruiting body, but it was releasing spores while I was making photos of it. This is what they look like on our 18th century laid paper. And we're looking at them from the top. So you see, again, the kind of like the fruiting body in the middle, heavily pigmented, and you can see the perithecial hyphae growing upward. And then in between them, this is kind of a bird's eye view, we see those thin little hyphae that are growing between the tops of the different fruiting bodies. And then with SEM, we are able to see with arrow number 2, you can see some of those rhizoidal rooting hyphae that are growing into the paper bulk. Next to the arrows number 1, you see some fruiting bodies with perithecial ornamented hyphae growing upwards and spores in between. And then arrow number 3 is pointing out one of the thin hyphae that are growing between the tops of these fruiting bodies. And this is zoomed in on one of the tops of the fruiting bodies. Number 2 are the spores of Chaetomium globosum. Here I'm getting a little bit ahead of myself. This is a photo after dry cleaning, and we're looking at the base of a fruiting body that was left on the paper. And you can see that it actually sits in a little pit. And we saw this in a lot of our samples, that they either grow into a little pit or they create one. And around it, you see some of the fruiting hyphae. And then this is with light microscopy. The fruiting bodies are still here. And I'm not sure that it's visible at this scale, but in this photo, you can see some of the hyphae that are following paper fiber surfaces in the paper bulk. The other species we worked with was Penicillium chrysogenum. Colonies of this species grow long, singular, erected spore bearers called conidiophores, forming a green, yellow to green-blue velvety layer on the paper substrate. The conidiophores branch into metulae and subsequently 3 to 6 phyalides, on which long strains of small spores are formed. So this is another sample that I took with sellotape. The strands of spores have been disturbed a little bit, and so they're scattered around, but they grow on these structures that we see at arrows 1 and 2 from the spore bearers. And this is what that looks like on paper. At arrow 1, you see some of those strands growing upwards. And then number 2, you can see some of those, again, those kind of higher hyphae that grow between the tops of these structures. And then with SEM, we see with arrow number 3, we see the hyphae growing on the paper surface. You can see that there's kind of different types of hyphae growing around, and number 1 shows some of those long strands of spores that are growing upwards from the paper. And this is what they look like up close. So we selected suit sponge, brush, and microfiber cloth for our dry cleaning tests because of their common use, but also because the differences in the way they pick up mold particles and dirt in general. A brush mostly just lifts particles from a surface. A suit sponge has a larger internal surface and they're somewhat sticky, helping both with cleanup, but also retaining whatever was picked up. And microfiber cloths, lastly, have a relatively large surface and have their static charge. All tools were applied with a gentle, even wipe, following the direction of the laid lines in the paper samples, and a clean new tool was used for each sample. There was less difference between the tools than I expected. The brush seemed to be least useful since it filled up with mold particles quickly and did not retain them as well as the other tools. We did not incorporate the use of a museum vacuum cleaner in this experiment, which would definitely be an important addition in a future research project. This is what the microfiber cloth looked like after cleaning samples with Chaetomium globosum. You can see kind of the spores spread throughout the cloth, and bigger structures, probably rooting bodies that are sitting deeper in the cloth. And then this is a sponge after using it on Penicillium chrysogenum. You can see how we kind of fragmented it, and it is colored green, and bigger structures and bundles of spores are found deeper in the sponge. So these are the results from our dry cleaning tests on paper. This is what the Chaetomium globosum paper looked like after cleaning. The thick and strong fruiting bodies of Chaetomium globosum were removed by the different cleaning tools, but left behind these visible black spots. And then that's an issue everyone who's cleaned a similar well-pigmented species from paper has probably come across. With optical microscopy and SEM, we can clearly see the remnants of fruiting bodies that seem to have been torn off rather than removed. And here we see, again, the bottom of what was a fruiting body still sitting in its little pit. And then on the right, we see rhizoidal hyphae remaining around one of those spots. And we can see spores kind of scattered over the paper surface. And with SEM, we noticed that the gelatin sizing showed a lot of cracks around the former fruiting bodies, kind of showing the force that we needed to remove them. And so here we're looking at the base of a fruiting body, and we're looking at kind of the rhizoidal hyphae that were tore off there. So around them, we see these burst rhizoidal hyphae, and scattered around are fragments of thicker hyphae, either rhizoidal or perithecial. In all samples, spores and clusters of spores are scattered over the paper surface, especially in deeper near surface areas, such as in between paper fibers, stuck behind a mineral filler, and in other indentations. And the parts of the mycelium that are attached to paper fiber surfaces are generally not removed. These hyphae are only damaged in spots where they are not directly attached to anything. And these are samples with Penicillium chrysogenum after cleaning. I left the bottom uncleaned to show the difference between clean and not clean areas. So these Samples looked clean after dry cleaning tests, and I would not recognize them as formerly mold-infested paper. However, with optical microscopy and SEM, it became clear what was left behind. The photo on the left here, again, the bottom is left unclean to show the difference. And the one on the right is, again, with light microscopy. In all samples after cleaning, a green color or accumulation of spores, strings of spores, and conidiophores is visible in indentations in the paper surface, mainly heaped up along the sides of paper fibers. In that sample, or that photo on the right, you can almost see the direction in which I cleaned this particular sample. On the surface, we still see a mycelium. Some hyphae are tore off where they are not attached directly to paper fibers. And here you can see, some of that mycelium that is left after cleaning, the spores. And if you look kind of in the pores of the paper, you can see little bundles of spores hiding there, where they probably like it best. And here you see that despite using a clean suit sponge and working in an aseptic mycological lab environment, probably the cleanest environment I've ever worked in, a cluster of Chaetomium globosum spores was actually left behind on one of these penicillium samples. So according to existing literature, superficial growth can be distinguished from fungal structures deeper in the paper bulk, and superficial growth should be relatively easy to remove. One can wonder to what extent we can speak of superficial growth on a porous substrate such as paper. We have not observed purely superficial growth in any of our samples of existing or newly cultivated mold growth. We can say the fungal structures become an integral part of the paper substrate during their growth. Without exception, the paper samples contained fungal structures after cleaning, whether they were visible with the naked eye or not. These structures continue to pose risks to collection materials and our health after cleaning and remain viable, ready to continue their growth whenever favorable conditions occur. We do not know how long a spore or other fragment will, with growth potential may remain viable on paper. So the work that we do is always about managing risks, and logically speaking, removing the majority of the fungal load from a collection item will decrease the risk of fungal growth in the future. However, I found it eye-opening to notice how much we leave behind and how we spread these fragments over and in the surface and near surface of the paper. I have been more aware of these effects in my dry cleaning, and not only when I'm working on mold-infested items. I'm aware that I cannot always tell the present fungal load with the naked eye. I'm aware that spreading of dirt and mold happens more easily than I can tell without using an SEM. This means I make sure to use clean dry cleaning tools and usually first work on the dirtiest parts of a surface before cleaning the rest of a surface with a new clean tool. Sponges and cloth are great at picking up mold and dirt structures, but also hold a higher load that can then be spread around. The golden question remains, when is an item clean enough? And remember, this goes for items that only receive dry cleaning treatment, but also for items that have been decontaminated with, for example, 70% ethanol or gamma radiation. Dead fungal structures may not be viable, but can still pose a serious health risk. Future research is needed to answer this question. How clean is clean enough? How viable are the fungal structures that are left behind? And when are we passing an acceptable threshold in our cleaning practices? To do so, we need more research on standardized protocols on how to grow mold on paper, which proved harder to do than expected, especially if you wish to create samples that resemble reality as much as possible. We need descriptions of the morphology and attachment of molds on paper and standardized protocols to evaluate mold removal during dry cleaning. Multidisciplinary collaboration between mycologists and conservators and conservation scientists is needed to gain a better understanding of favorable conditions for mold growth on paper, how different growth conditions influence the morphology and excretions of fungal growth, to which degree lab cultivated mold samples represent actual mold growth in paper-based collections, and lastly, to gain a better understanding of the degradation of mold structures and excretions after a growth phase. This is not a phenomenon that is generally studied in mycology, since they usually look at fresh, new mold growth, but it is a situation that we come across a lot, for example, when formerly infested materials enter our collections. So I really want to thank everyone for joining in today. And here are some key references I will share through CCHA, a handout to everyone who is here today with more literature to dive deeper into this topic. And I want to thank all these people for helping out with the research. Please see that my email is here. I'm looking forward to everyone's questions. But if we don't get to them, or you have thoughts or recommendations or questions later on, I'll be really happy to hear from you. Thank you so much.
Thank you so much, Fleur. That was just incredible. I feel so much, I feel so full of information right now, and I'm sure everyone has a lot of questions that are going to be coming in as well. I have an initial small question in the chat of why would ethanol be preferable to isopropyl alcohol for paper?
That's a good question. I've actually mainly worked with ethanol when it came to mold. I do know that ethanol, when not used in a 70% solution, can actually kind of activate mold. So if you're using a lower ratio or even a higher one, such as 90% ethanol, that can actually activate a spore. It can kind of kick them into a survival mode of where they shut down and protect themselves and can be viable still in future. So I don't really know what the effects of isopropanol are in that regard. But yeah, molds are pretty specific to how they respond to the cleaning solutions that we use on paper.
That's a really good point. I have a question about one of your slides where you showed how the gelatin sizing cracked around the fruiting body. And I wasn't quite clear on, do you think that that happened due to the fungal growth or due to the dry cleaning?
Yeah, they seem to, let me go back to that one real quick. There it is. We mainly saw this cracking in samples after dry cleaning. Or that's where we noticed it most. It could be that they're hidden before that. And we definitely saw that the rhizoidal hyphae are kind of like pushing paper fibers aside as they grow into the substrate. But you do, and that's, I think, pretty typical for these types of species with kind of like these thicker, well-pigmented structures, kind of stronger structures, is that you actually feel resistance when you're cleaning these items. And so, yeah, it seemed that we were actually kind of damaging the surface right around these structures as we were cleaning. Does that make sense?
I completely believe that's happening because I agree with you about the resistance you feel sometimes. So it's really hanging on, isn't it?
Yeah.
I have a question in the chat, which I hope that Nicole is referring to not collection objects. She says she's tried a tea tree oil and deionized water solution to clean her mold outbreaks. And have you tried or heard of this?
I have heard of them. Let me see if I have this article here. No, but I'll have it in my longer list. There's a really good article on different types of cleaning solutions and different types of treatments. But I have personally not worked with these methods. No.
That makes sense. And I see two very similar questions about using a vacuum cleaner, which you've already mentioned would be good to look into. Do you have any plans to further your research? And do you anticipate that doing two types of cleaning, both dry cleaning and vacuuming, would offer benefits?
Yeah, I think that, you know, a vacuum cleaner helps with the fungal load that builds up in your brush, and also just helps to kind of remove whatever you're picking up. And hopefully less of it settles back down on the paper surface. That would make sense. I did have plans to continue this research when I was living in Holland, but I moved to the US a few years ago, and life kind of happened. And I'd be really happy to continue this at some point. But currently my current job keeps me pretty busy. But yeah, I really, you know, I hope that there are students or others out there who can jump in on this and you know, might be able to kind of extend this experiment that we did. And I'd be really happy to share, you know, the methods that we used and the things that we discovered about, yeah, like I said, growing molds on paper, which wasn't as easy as we thought it would be, which surprised me. And to, you know, to make samples that actually look like what fungal growth looks like in reality on our collection items. So yeah, I'm really hoping that at some point we can continue this in some form or way or shape. And I think including a museum vacuum cleaner will be a very logical next step.
Yeah. And you mentioned a couple times that this can be done in combination with sterilization steps, including gamma radiation. How frequently do you think this is used on collection materials?
Yeah, this has been interesting about moving from Europe to the US. I think there's a small difference there in maybe, what is, maybe it's a kind of a cultural difference in the conservation world. Gamma radiation seems to happen in Holland more often than here in the US. And I think collections are kind of doing it less than they might have, you know, a decade or a while ago. But it's still pretty common, especially after mold growth has been found. It's something that is used generally. And yeah, it seems less common here. But these are things that I'm still kind of discovering after moving here. Like what is more common here, and what happens more often here. So in Holland, we're used to, you know, sending out a pellet with a bunch of boxes to get them treated, and then they get cleaned afterwards. And here that seems to happen less often. And I think that's a choice. It's a, you know, weighing of risks that seems to be a little bit different here in the US.
I appreciate that perspective. I know at CCHA, we've sent things out for gamma on very rare occasions, and usually more when it had other contaminants, like sort of rodent droppings, that we were more concerned about other sort of biological things as well. And also for lower value collections, I'd say. So it's pretty rare for us.
Yeah, that makes sense. I do want to say that is kind of my personal observation. This is not something it would be interesting to kind of see the numbers and what the differences are in treatments.
Yeah, we certainly found that having our Dutch fellow that we could observe some of the differences in just what we consider normal versus what's normal in Europe as well. Someone asked about to clarify whether your results are from cleaning active or inactive mold, and would they be similar?
Yeah, so our samples had been like the mold had been grown shortly before the dry cleaning tests. And we did let them dry out a little bit before the dry cleaning. And so in that respect, they were maybe not as actively growing as they had been some days earlier, but they were very close to their active growth phase. Yeah, one thing that I found surprising is, I worked a lot with Jos Houbrake, who leads the research group on indoor fungi at Westerdijk. And they generally do not study mold structures long after their growth phase. And that's kind of the nature of the type of research that happens in mycology. A lot of mycologists are, especially indoor mycology, is focused on food production and food storage. Mm-hmm. And so, yeah, typically they look at, pretty active mold growth. And that was pretty surprising to me that, I went back to the Dutch Cultural Heritage Agency. That's where we worked with our scanning electron microscope. And I took those photos back to Jos, expecting that he would be able to explain to me what we were looking at. And sometimes we actually had a hard time doing that because they typically don't look at very old mold structures that have grown, especially those samples that we took from Dutch collections. Some of them might be years or decades old. And so, yeah, that's kind of a whole new terrain in mycology too, to look at these degradative mold structures.
That's interesting, and that's part of the interdisciplinary nature of your project, that we're seeing how the other fields interact with mold as well. As a future recommendation, would you consider it worthwhile to isolate those pages even after cleaning forever once they've been infected?
Yeah, that's an interesting question. Like, how clean is clean enough? When is something clean enough to place back in the materials in storage? When is something clean enough to send out to the reading room? Or, use in any other way that we use our collections? I find that a really hard question, and especially after I started working here with the special collections, I get confronted with it, more often than I thought I maybe would. Yeah, we recently had a collection with old mold growth that has been cleaned, and still, I'm not sure if I would feel comfortable placing it with other collection materials and sending it out to the reading room. This is a really important question that I think we all are very familiar with, and that is really hard to answer. More research needed.
Yes, and I see lots more questions about vacuums, and I think you've talked about.
I do want to say one more thing about the how clean is clean enough. I would definitely be interested to hear thoughts about this from other collection managers or, conservators who work with collections as well. How do you deal with that in your daily work? And, when do you determine, when do you feel like something is clean enough to use in a normal way and to store in a normal way?
I see a question about having a copy of the question and the answers, and this part is still being recorded, so it will be included in the videos that everyone receives. Could you repeat the name of the standards that you referred to for treating mold?
The standards.
That's
Sure.
maybe Stephanie can come back with a little more detail on that because I can't say I caught that either. Let's see. I see a lot of thank yous and appreciation, which is not the same as. Oh, someone is asking if Groomstick was considered. Are you familiar with that product? It's very tacky, sort of kneadable. Yeah.
I'm familiar with it. I've actually never used it. I think it's a, yeah, kind of like sticky.
Mm-hmm.
You can turn it into a ball and kind of roll it around. From what I understand, it leaves quite a bit of residue on the paper. And it leaves some of its like residual stickiness. And so I've actually never used it in my practice. I don't know if you all use it at CCHA or what your experiences with it are.
I agree with you. We no longer use it because of that concern about residues, but there is something that we use that works in a similar way for very, very local spot reduction, especially on pastels, is dipping little sticks into Lasco and letting it dry. And that has just a slightly tacky surface, but it's sort of a conservation adhesive that we're familiar and comfortable with. Let's see, everyone loved your picture. All of your pictures are just amazing.
Yeah, I love the Let's Go idea. That's really, I don't know, I like the like creative use of the materials that we have around.
Oh, this is a great idea. Did you look at any papers that you thought were unaffected by mold under SEM to see if there was inactive mold that we just hadn't perceived yet?
Yeah, that's kind of what you start wondering about, right? We when we were looking, so the papers, okay, so the answer first is no. We looked at areas of pages where, you know, other areas were clearly affected, and we took samples from areas that seemed to not have been affected, but they were, you know, on the same page and in the same item. The paper that we used for this experiment came from the research or reference collection from the University of Amsterdam of historical papers. And we looked at all these papers before I made a selection of possible papers to work with, and we looked at them with SEM before we did our experiment to kind of understand what materials we were working with and if there was anything present before we started growing anything. And we did find there was one paper that seemed to be an old album page that had that we found arsenic on. So it did make me realize there, you know, there's papers where you have no idea what might be on or in them as you're handling them. And I think that's something in general to be aware of. But the paper that we used seemed to be pretty mold-free before we worked with it, even though it had been around for centuries. We did have some bacteria that started growing in some of our samples as well. I think there was actually one photo in my presentation that had some hidden bacterial growth somewhere in a corner, because sometimes it's really hard to avoid when you stick them into a warm and humid environment. Yeah, so there's much more present than we're usually aware of.
It's so interesting how much more you can see as soon as you start looking. You find more and more information. And a related question, how does the presence of other potentially damaging materials, such as fecal matter, rust, water damage, affect how you would clean the paper artifact or the dry cleaning process?
Man, I would kind of want to ask that person to elaborate on that question.
That could be a 2-hour answer.
Could you repeat it? I can also open the chat.
Let's see. How does the presence of other potentially damaging material, such as fecal matter, rust, or water damage, affect how you clean paper artifacts?
Yeah, that's a whole topic in and of itself, I guess, or a whole lecture in and of itself.
The
Yeah,
answer may be with care, right?
yeah, tread with care. Yeah, like I said, I guess, generally in dry cleaning, I always start with the things that seem the dirtiest or the easiest to spread, or and I kind of go from there. Does that kind of make sense?
Mhm. Yeah. And then there's a question from sort of a layperson who's like, now that you've shown us all the mold that's remaining, is there any way to actually kill the mold?
Yeah, so there are ways to kill the mold. The gamma radiation that we talked about earlier usually has a pretty high success rate. And yeah, there are methods with organic solvents or other materials that are used, and a range of other options, but those are the ones I think that are used most often to kill mold structures. One important thing that I do want to point out, and I kind of did in my presentation, is that even after mold structures have been killed or made non-viable, they do still pose a health risk. So they can still cause allergic reactions and pretty severe allergic reactions and other illnesses. They create mycotoxins that can still work after the mold doesn't, is dead. And so that is something to always be aware of when you're dealing with moldy materials. And they can also still cause degradation to the materials that we find them on and around. So yeah, even if we kill mold structures, we still want to lower the fungal load as much as possible by dry cleaning.
That's a great, very complete answer. There's a question about, would you recommend to individually box an item after it's cleaned to help mitigate any spread of remaining mold?
Yeah, I guess this is so context dependent. And so dependent on the type of material and the type of collection and storage that you have, after you've cleaned something, and if you store it under, stable climate conditions, and you take good care of it, and there's not a lot of residual mold left on the paper, yeah, it's a risk management story. So you're removing a lot of the fungal load, but there will be some left. And so if you store it under stable conditions, it should be okay. But it really depends on what it looks like, what your material looks like, what your storage space and your organization looks like. I think it depends on a lot of different factors. But it could be a smart move.
And we're just hitting 1 o'clock, so I'm going to call this the last question of, do you recommend a fume hood when you're cleaning mold from paper?
Yeah, so fume hoods are often used, but I think you'll call it a biological or decontamination cabinet is actually, I think that's what it's called here, right? That is actually a different type of environment. So the problem with a fume hood is that you can still, depending on where the air goes and how it's filtered, you can still spread particles that you are removing. And that's something to really be aware of. Fume hoods are not made to work with molds. You really need a specific biological cabinet to work with molds. For example, we don't have that here. We just opened a preservation studio last year, and Yeah, so I know that I'm not able to work on moldy materials here. I isolate them, and I'm still working on ways to deal with them after we find them. I've worked in another institution where we had at some point set up a table outside to work on moldy materials because that was possible there. And we're about to empty a former offsite storage that the University of Arizona Special Collections has. And there we will also set up a table outside. That's how we'll clean our materials. These are not necessarily moldy materials, but they're very dusty. And so, just to be as clean as possible, that's our solution here. But yeah, fume hoods are not made for biological hazard materials. Yeah.
Yes, we've been researching upgrading our fume hoods, and we've been learning about, yes, and just because mold is a particle versus a gas that you're trying to trap as well. But definitely that's what we recommend for small collections who we would never expect to own a fume hood, is to work outside if they can, for the same reasons. So I would like to thank you again for this incredible presentation. You're getting so much appreciation in the comments. I hope you take a quick look at that to see how much everyone here has loved hearing about this and getting to see your pictures and benefit from your expertise. I'll remind everyone who's here that we will be having a quick little survey after this closes to help us shape our future programming. And thank you everyone for attending as well.
Thank you.